Okay, today we're transitioning away from very molecular and cellular aspects, primarily molecular aspects underlying neuron physiology, and we're building up from there. So we're kind of moving up to more circuit level aspects of how the nervous system works. And I've mentioned this a few times about the fact that we have sensory input that comes into the brain. It is processed and analyzed by the brain. Decisions are made, memories are formed, but eventually what we see is some sort of motor output, right? It results in a change or some kind of activity that we can see as the brain acting on the muscles ultimately. And so that's kind of a real simple rubric of what brains do. For the next several lectures, we're going to be talking about sensory input, and what better way to start than talking about eyes? So first, we got to talk about what are eyes used for. So it's for sensing light, and so I'm not going to dig too deep into the physics of light because presumably you've taken a physics class or you will, and you'll get more information about precisely how the physics of light there. But there are a few things that I need to mention, of course, right? So light is referred to as an electromagnetic type of energy, and it can be seen and described as having a wavelength and an amplitude. The wavelength will determine where it falls along the electromagnetic spectrum, and the amplitude refers to how intense the light is, right? The range of the electromagnetic spectrum can go all the way from gamma rays with very, very short wavelengths all the way out to like broadcast bands, AC circuits, much longer wavelengths. The band that we see is called visible light, and it's actually a relatively narrow band of the available electromagnetic spectra. There are animals that can see into ultraviolet and infrared. This is what humans can generally see. So this is the visible wavelength spectrum, and it ranges from violet up to red. And it usually ranges from a little lower than 400 nanometers in wavelength. So that would be, from trough to trough or peak to peak. And it can go up into the deeper reds, which is around 700 nanometers in wavelength. And then everything in between are all the colors of the rainbow. Okay. So as far as the visual system is concerned, this is what we're going to be discussing today and tomorrow. It's a fascinating organization, the way that the nervous system can take in this light information and and pull lots of detail about the world into our brains and the way it gets processed. It's a super fascinating process. I really wish we had more than just two lectures to talk about it all. And we'll get into more of the details of all of this. But I'm only showing this because partly I'm mostly focused on the way the visual system is organized in humans and other non-human primates. We will talk about cats to some degree next time, but mostly we're talking about humans. And so the way the visual system is organized in all terrestrial vertebrates is essentially the same, but there are differences. A lot of it having to do with whether we are binocular, where our eyes are pointed forward, where both eyes are used to sort of see the world in almost the same perspective, but slightly different perspectives, versus monocular, where the eyes are largely oriented either like to the sides is generally how it is, and where you have a greater range of view that you could potentially see, but you don't have a whole lot of overlap in your visual field. But here we can see, like humans and other animals that have binocular vision, that there's a lot of overlap that each eye can see. Okay, so let's talk a little bit about the eye. All right, so the eye is what we're going to be focusing on today. This is a schematic of the basics of eye anatomy. And yeah, so we're going to go through the basic parts. Most of what we're going to be talking about today is the retina. The retina is the only place in the eye that has light sensitive cells that gets transmitted and eventually generating action potentials that can be read by the brain. The Another important part is the ciliary body. So these are muscles that encircle the lens, and they can change the shape of the lens. The iris is the colored portion of the eye. It has two sets of muscles that can control the size of the pupil, which is the hole that light is passed through. So the pupil can change its size, and that will control how much light is getting into the eye. Right. So if we're looking at a very bright visual field, the iris will close more, and therefore the pupil will be smaller, and that will allow in less light because we don't need so much light. The retina is going to have a range at which it works best to see detail and color. And then if we're in a very dark field, the iris will open up quite a bit, and then the pupil will be bigger, which will allow in more light for us to pick up more detail. Sclera is just the white out part of the eye. So if you were to take the eye out and look at it, you would see the white of the eye, and that's called the sclera, which is just a tough outer membrane that holds the eye in place. And then we have the cornea, which is a transparent tissue that forms the outer layer of the eye on the front, and it permits light to flow through the eye. So the cornea is going to be in here. Okay. So the lens, though, is the thing that actually shapes the rays of the light that is coming in that gives us focus, which we're going to talk about in a couple more slides. But one thing I want to talk about is that when we are talking about what an eye can see, is that we're talking about the visual field. What is in front of us gets taken into the eye. And that visual field for a typical human eye has the ability to see around 150 degrees of space. So if we're just staring at a point, each eye can take in around 150 degrees. And you can see that if this is the right eye, obviously we can't see everything over to the left, but we have a little bit more to the right that we can see. And then the left is just the mirror image of that, because the eyes have a mirror image orientation to each other. And so if we're looking at an object, say a pencil, what happens is, because of properties of light, which I'm going to assume many of you know, and we'll cover a little bit into detail, but they pass through the lens, and so these photons are coming from the tip of the pencil, they hit the cornea, they pass through the cornea, they hit the lens, and then because the lens has a certain shape, there's refraction that occurs, and it actually reverses the image. And so the tip will pass here and come to the if this is the right eye, this is referred to as the temporal side of the eye. If the tip of the pencil is faced more towards our left, okay, from our perspective. So temporal refers to this side, and then the eraser over here is going to have photons coming off. They'll be largely red in their orientation, sort of pinkish hue. They will pass through the lens, and then that gets transposed and flipped, right? Because that's just the nature of the way lenses work. And so it'll be on the nasal side of the eye, which is where our nose is. So we have a nasal side and a temporal side. In the right eye, the temporal side is going to be to the right side of the eye. And then for the left eye, it's going to be the reverse, right? It's a mirror image, right? So, which is important, especially for what we're going to talk about on Wednesday. So to focus, generally what we need is that if we're looking at something in the far distance, we don't need a whole ton of refraction. Refraction is the term that we use to how we can bend light. And because the lens is actually a malleable structure, it can change its shape. Those muscles in the ciliary body can either push it or relax and then let it sort of flatten out. If it is flat, then we have less refraction. If you can think of the way light just passes through flat glass, like it doesn't refract at all. But if it's slightly curved, it refracts to some, and that's what we're talking about here. And so it doesn't actually need a whole ton of refraction in order for us to get a sharp image on the retina. So we're trying to project the image onto the back of the eye where the retina is located, and so we don't need a ton of refraction. Now, when we are, that should say seeing near, sorry, but what we need is a round lens. So if we're looking near, so the object is pretty close to us, what happens then is the ciliary bodies, they contract, and then they push the lens to be more rounded, and it's fat in its shape. So then it has more curvature, therefore light refracts more, and therefore we get an image that gets then transposed to the back of the retina that's in focus. And this is how we can change focus Whether we're looking at far objects or close objects, it's the ciliary muscles that are changing the shape of the lens that allows us to do that. And it happens very rapidly. We don't even really think about it, right? Just almost instantaneous. As you're flitting from looking at me to your notes, it's in focus for hopefully all of you, and it almost happens instantaneously. So some of us, such as myself, we have glasses, and that's because our some aspects of our eye don't work as well as we would like, right? So ideally, what would happen is that we get, say, if we got rays coming into the eye, and then it should converge into a point, or at least creating an image. Like if we have some sort of pencil that we're looking at, we would imagine the pencil would be transposed in reverse orientation, top flip to bottom, left flip to right. So it's a mirror. It's flipped, but it would be in focus, where all the rays of light are transposed onto the back of the retina, and they're in focus and they're not fuzzy. If they are out of focus, say for instance, if we have something called hyperopia, which is farsightedness, it means that you cannot see objects that are very close. So that's what it means to be farsighted. So you can see far, but you can't see things very close. What happens there is that if you're looking at an object, say this painting of this beautiful flower, if you suffer from hyperopia, what happens then is that the image would actually be more in focus past your eye. And so it means that you can't get enough curvature into your lens. That's one way that you can get hyperopia. And so because that means you can't refract that light so that it actually is in focus on the back of the retina. Instead, you can see that the rays of light are not converging onto the retina. They are, they would converge on the behind the eye, but of course, there's the sclera, and there's no light-sensitive cells there, so it just shows up as out of focus. Does that make sense? And we can fix that by having a curved lens that then accommodates that, and so then we get some refraction before it gets to the lens that's inside our eye, so that we get normal in focus light that is now where it should be in the back of the eye. And so when you go to the eye doctor, check your eyes, and then they'll get you the right size lens that you need to get the right in focus. So, and then myopia is nearsightedness. Okay, so that means you can see things that are close, but you can't see things that are far away. And so for this, what is happening, it's basically the opposite. So we can't flatten our lens very well. And so what happens then is that we have sort of a fatter lens than it should be. That means the light coming from any sort of object that we're looking at actually converges ahead of the retina. So there would be an in-focus image, but it's not landing on the retina where it should be, it's landing in front. And so, of course, what that means, by the time the light gets to the retina, it is now out of focus, because the light needs to be precisely converging at exactly the right place. Okay? And so the way we fix that is with some sort of convex lens that refracts the light outwards to go through our sort of fat lens, so that we now can push the image back so that it lays flat on the back of the eye. Okay? So, and then LASIK is one way to fix conditions like this. I've got a little video at the end. If you're curious to see how LASIK works, you can look at that little video. But I figured there just wasn't enough time to cover that. All right. Questions on that before we go on to talk about the retina? That's basically what I'm going to say about light and how it's hitting the retina. So the retina is very thin, covers much of the back of the eye. And if we were to look at a cross-section of it, we would see layers of cells. Okay? And so this is a zoomed-in view of what that looks like. And so we're going to be zooming in even further and talking about what these layers do and how it works. So what we're looking at here is now an orientation where the light is coming from the bottom. So I've taken that little chunk and then we turned it 90 degrees. So this would be the back of the eye here. This would be the surface that's facing to the inside of the eye. And light is coming through here, okay? So there are three basic layers of cells that are arranged inside the retina, and believe it or not, they're arranged inside out. So light actually has to pass all the way through all these surfaces, and the rods and the cones, which is obviously, most of you probably know this, that these are the photosensitive cells that are within the retina that convert light information into chemical information that can be read by the nervous system. They're located in the back of the eye. So light is going to pass through and hit those rods and cones. The next layer are bipolar cells, horizontal cells, amacrine cells. There are some other cells too, but those are some of the main ones. We won't be talking about all of them, but we will be talking about bipolar and horizontal cells. These are local circuit neurons that sort of take information from the rods and cones and then pass them on to the next layer, which is called ganglion cells. These are projection neurons that form the layer closest to the inner membrane. Now, ganglion cells are the only cells in the retina that are true neurons. They actually have action potentials. These other cells are nervous system cells, and they do fluctuate their membrane potential. They release glutamate, some release GABA. Some of them are neurons, but many of these, some of the ones that we're going to be talking about, don't really act like neurons. And so I don't like to refer to them as neurons, they're more like cells. And so we'll get into that. So when visual information is entering the eye, it strikes the retina to begin processing. So that's the first step. So as light is coming through, we have light, it passes through all of these layers, and then it ends up striking either rods, cones, or a mix of the two, okay? So if we've got light passing through, it's going to pass through these layers, and it has to hit these cells first, because the vertebrate eye is built inside out. It ends up deactivating photoreceptors. So we'll get into the chemistry of that in a moment. Rods and cones, they transduce energy into electrical energy. So they transduce light energy into electrical energy, electrochemical energy, I should say. And the rods and cones and photoreceptors, they synapse onto bipolar cells, which is going to be what we're going to be focusing on as far as the circuit goes. So those bipolar cells then synapse onto ganglion cells. And then the ganglion cell axons, they go through the optic, they transmit their information through the optic nerve, providing that visual signal to the brain. And this is the primary neuron in the visual system, in the visual pathway, the very first neuron. Okay, so we refer to that as the primary neuron. So we've got receptor cells, we've got, which are going to be the photoreceptors, we've got these bipolar cells, which help with processing information, which we're going to get into. And then we've got ganglion cells that take that information and then transmit that to the brain. Okay, so let's talk about photoreceptors a little bit. So rods and cones, these are the two main types of photoreceptors that we can find in the eye. And they differ in many ways, but they're actually very similar in a lot of ways. So we're going to focus a lot on their similarities, and we're going to talk about some of the differences. So they both use similar molecular machinery to convert light to neural signals. So it's very similar, at least the basic molecular machinery. And they both exhibit the same general structure in that they both have an outer segment like this, which has all of these discs. So if you were to look at them, they would look like a series of discs. They have an inner segment that contains the cell body, the nucleus, mitochondria. And then they have a synaptic terminal that's filled with synaptic vesicles containing glutamate. So they both release glutamate. Those are the similarities of the photoreceptors, rods and cones. There are some differences. So rods have a photopigment called rhodopsin. They have a high concentration of this photopigment. Whereas cones contain one of three different photopigments called the opsins. And this is because cones are the ones that contribute to color information, which I'll get to in a little bit. Whereas rods tell us more about intensity of the light, the brightness of light. They do other things too, which we'll get into. So rods, they respond to low light conditions. So they're particularly sensitive to low light conditions. That's where we're going to see majority of the activity of rods when we're in low light condition. But they don't distinguish colors at all. Whereas cones, they tend to operate under bright light conditions. They require at least 100 photons striking the outer segment in order for a given cell to respond. And as I said, they permit for color vision. There's a high level of convergence for the rods, which means that they provide little spatial resolution. I've got some slides that explain this in a little bit more detail. Whereas cones typically have very little convergence. Typically what happens is that you have a single cone will usually be connected to just a single bipolar cell, whereas rods, there's a lot more convergence. And I've got a couple of slides that explain that in a little bit more detail. And then People that have problems with their rod function, they usually have difficulty seeing at night. So that's one reason why someone might have problems seeing at night. And then people who lose cone function, if they lose cone function at all, they're functionally blind. So if they have no cones at all, they cannot actually see at all. So you can lose most of your rod, the rods, and you could still more or less see okay, but you would have a lot of problems with some of your peripheral vision and especially vision at night. But if you lose your cones, like all of your cones don't work, you're legally blind. You wouldn't be able to see anything. So cones obviously play a very big role. Rods are also important as well. So let's talk about the opsons versus the rhodopsins. So let's first talk about opsons. The reason why cones can encode color vision is because they have 3 different opsons that are sensitive to, that have peak sensitivity to particular wavelengths. So these, we have blue cones, we've got green cones, and we've got red cones. And what that means is that there is a peak wavelength that is most optimal in eliciting activity in those opsons. So for blue cones, it's going to be blue light that elicits those opsons to fire. Whereas green cones are going to be more green light, around 534 nanometers, and red cones are going to be sensitive to red light, which is around 564 nanometers. Okay, and so it's the combination of those things, which I can't get into at all, unfortunately, to talk about how we encode color vision because it's just too much to cover. But it is that combination of information that the nervous system takes advantage of in order for us to understand color. Rods, they also have a wavelength that they're most sensitive to, which is 498 nanometers. But again, they do not use. We don't use rod information to encode color information. We use it primarily for distinguishing the contrast of objects and intensity of light, and it provides a lot of information about what's happening in our peripheral nervous system. Okay, in the peripheral field of view, I should say. So some of the sensitivity of intensity of light is we're going to be going over here. So the way we talk about intensity of light, we refer to that as luminescence, and this is essentially like a log scale. So a log scale means it's a power of 10. So we're talking about a wide range of intensity that we're talking about here, even though we're going from -6, -4, -2. It's actually a wide range of numbers of photons. So the first kind of vision we can talk about is scotopic vision. This is where we have low light levels, and it's during these phases under scotopic vision where the light is so little that we get very little to no activation of cones. So it's almost entirely rods that are going to be active if we're talking about this low light vision. And this is typically going to be like starlight type levels of intensity. So like on a moonless night, and all you have out there are just stars, basically. Believe it or not, you can still see some things, and that's going to be entirely rod information that's giving you that information. So the next is photopic vision that I want to talk about. So photopic is going to be in this range here. And basically, at this point, the rods are largely saturated. So they are no longer really contributing much information to the visual field that we're seeing. This is going to be anywhere from like indoor lighting to sunlight. I mean, they're contributing some information, but they're not dominating like our visual information. And then it's in this case where we have the cones. So most of the information that we're getting here is almost entirely dominated by cones. So it's cones that are going to have this other set of sensitivity. Like I said, they require more photons to be activated or deactivated, and as we'll get to, and that's what allows them to provide the information that's needed. And then we got mesopic. Okay, meso means in the middle, and that's going to be somewhere in between. And that's when we have both rods and cones contributing. And the light condition for that is relatively dim. So like a dim room that's very sort of dark, maybe candlelight or moonlight. We have both rod and cone information that's providing information. And it's not as if there's like a hard cutoff. It's like a gradient And this is just the overlap of where like rods become less and less important and dominating the visual field, whereas cones become more and more important until we get up to a place where they're almost fully dominant. Does that make sense? Okay. So one thing that's interesting about the way cones work, photoreceptors work, is that if we're able to measure the membrane potential, so if we flash a light, right? So as I said, these are light-sensitive cells, and all we need is just like a tiny little flash of light, and we can strike a cell, and then we can measure the activity. We can change the intensity of the light, so this would be one flash, but we could make it higher and then higher and higher, so we just increase the intensity of that light. If we do that over a number of trials, this is the response that we see. So we see it seems like these cells are being held at around negative 40 millivolts, and that as we increase the intensity, we get hyperpolarization of these cells. So by striking these cells with light, they are hyperpolarizing, which is interesting. The other thing we can note here is that we have that it's a graded response, right? So low intensity results in some change in membrane potential, but it's small and not as long lasting. But the higher intensity, the more we increase the intensity of the light, we see a greater and greater response, as well as the length of the response lasting longer and longer. So you may have noticed this if you've ever looked at the sun briefly, and it's like you close your eyes because it hurts, and then you can still see the intense action of the sun like in your eye, even though you're not even looking at it, but it's kind of like, oh my God, what did I look at? That's some of this lingering effect. You've bombarded the retina with way too many photons, and you have a long-lasting hyperpolarization. And remember, we were talking about action potentials acting on the order of milliseconds, no more than tens of milliseconds for sure, right? The EPSPs, they last 2 to 3 milliseconds. The time scale here is hundreds of milliseconds, right? So with each individual cone or rod, essentially we have a longer lasting effect. It's still short, right? Still less than a second, but like this speaks to some of the chemistry that's happening. So let's talk about that chemistry a little bit. So what happens is that when light comes in, and in this case we're talking about a cone, a rod, but it essentially works the same way in a cone. In rods we have rhodopsin. Rhodopsin is the protein that is light sensitive. So we've got a gene that makes rhodopsin. It gets turned on. It gets incorporated into the discs. So here we are. We're zoomed in at the end of a rhodopsin. So we're looking like of a rod. So we're looking right here, and we're looking at the first disc. And the disc is made up of a membrane, and inside that membrane is loaded with these rhodopsins. Okay, so there's a bunch of these rhodopsins here. This is going to be the light-sensitive part of the cell. And so when light strikes the rhodopsin, it then causes it basically acts this protein called transducin, which then activates a phosphodiesterase, okay, which then hydrolyzes cyclic GMP. So it takes cyclic GMP and converts it back to GMP. Now, cyclic GMP, when it's in high concentration, it tends to open up sodium channels. These channels that allow sodium and allowing for some calcium to flow through. These channels, if they're open, we've got current flowing through. Now, what happens is that when PDE is activated, this cyclic GMP gets converted to GMP. It becomes hydrolyzed. So then this becomes disassociated from the open channel, and it needs the cyclic GMP bound in order to be open. So what ends up happening then is that the channel closes. And so now we have no longer any of this current flowing through, which means that it becomes hyperpolarized, right? Sort of like how a neuron is, like if it's got closed sodium channels, we are not having positive current flow in, which means that it is now going to decrease its overall membrane potential, right? So, ironically, light deactivates a cell. That's what I mean by deactivate. So it's lowering the membrane potential. It's making it hyperpolarized, okay? Does that make sense? Okay. So, in dark, basically what's happening is that we've got a lot of cyclic GMP present within the rod in the outer segment. that cyclic GMP is open to these channels. We've got calcium and sodium flowing through. Mostly it's calcium, and mostly it's sodium that's driving this current. And then the membrane potential is relatively high. So it's more depolarized than hyperpolarized. When light hits, then we've got the rhodopsin that gets activated, transducin gets activated, the PDE gets activated. It converts cyclic GMP into GMP. That means that the cyclic GMP can no longer bind to these channels, leaving them open. So therefore, we've got reduced calcium and sodium current going into the cell. The only current that's open are the leaky potassium channels. And therefore, and that's flowing out of the cell because the inside of the cell is high in potassium, it's low on the outside, and therefore we've got a hyperpolarization event. So it hyperpolarizes, it decreases its membrane potential. Okay, and the thing about this is that it's not an action potential, right? So its membrane potential is a lot more like a speedometer, which is kind of what we're illustrating here. So it goes either up or down depending upon how much light it's hitting. So if it's less light, the membrane potential goes up. It's almost as if it's going faster. So it's becoming more and more depolarized, which means it is releasing more and more glutamate. But then it goes down if you hit more and more light, and therefore it becomes hyperpolarized, and then it releases less glutamate at its synapse. Does that make sense? But it's not. It's also releasing this glutamate constitutively. So it's not releasing glutamate in a controlled way, the way an action potential is. That's why these things are not neurons. So if it is more hyperpolarized, it is releasing less glutamate. If it is depolarized, it releases more glutamate, but this is graded. It's not all or none like an action potential is. Does that make sense? That's why this isn't like a neuron. Okay. So I mentioned convergence. This is what I mean by convergence. So rods, typically the way that they get organized is that because what I'll talk about in a moment on the way that they're located within the retina, what often happens is that you have many, many rods talking to one bipolar cell, which means that this bipolar cell is collecting the light information of lots and lots of rods. And it tends to be clustered into a particular place within the retina. So it's going to be a certain space that's going to have somewhere between 10, 15, maybe as many as 30 rods. All that information is going to converge onto one bipolar cell. Okay? So that one bipolar cell is now taking in the information of like a bunch of rods. Whereas cones, they have very little convergence. So there are some places, especially on the outer parts of the retina, where you can have a few cones that may converge onto a single bipolar cell, but especially as you get closer and closer to the center of our field of view, that's where we see single cones going to a single bipolar cell. And so what this means is that if you think about this, if you've got light information coming into the retina, part of the reason why our rods are so sensitive to picking up low intensity light is because you may have only a few photons that are hitting the retina here or here, but that information can be converged and collected into a single bipolar cell. So that single bipolar cell can take some of the change in stimulation for some of the rods, and then it may induce a change in the membrane potential of the bipolar cell that'll cause it to change its activity. Whereas here, the single cone, it can only see light that is hitting it. It can't see light that's going here and here and here, right? And therefore its resolution is much finer, whereas this has much wider range of resolution. So it can pick up light from a wider range. Does that make sense? So that's one of the reasons why rods are sensitive. Part of it is also because they have a lot more photopigment. The outer segment in rods is much bigger than it is in cones, but also this convergence mechanism is the other thing. So it doesn't provide a lot of detail for information because this bipolar cell doesn't necessarily know if the light is here or here. It's just sensing, oh, there's light, and therefore there's light somewhere in this receptive field, and that's all that it can see, but it can't see if it's here or here. Because ultimately, we're going to see a change in memory potential in the bipolar cell and it doesn't matter if it's over here or here. Whereas here, there is very specific information about precisely where in the retina the light is hitting, because it's only this cone that's driving this bipolar cell. Okay, so its receptive field is much smaller than this. All right. So the other way to look at the retina, right? We've been looking at the cross section, but we can also look at it's a sheet that's on the back of the eye. And so if we were to look through the lens, and if you go to an eye doctor, they can look at the what your retina looks like. So instead of looking at this thin sheet, this is the retina here, right? We can look at the back surface of the eye, and we're going to see a few things. First of all, we're going to see something called the optic disc. Okay, so the optic disc is also referred to as the blind spot. And I'd like to talk more about the blind spot. I'll talk a little bit about it, but it basically doesn't have any photoreceptors in that spot. So we have this little spot that our brain sort of fills in with information of nearby photoreceptors. But if there's any detailed information in a tiny little spot, we won't be able to see it. That's called the blind spot. And it's off to the side of the center of our field of view, so it doesn't matter that much. Our brain doesn't even notice it. It just fills in the gap. But the reason why we have a gap there is because we have all these retinal ganglion cells that are on the outer surface, and they have to carry that information out of the eye, and that has to be carried through the eye in the back to the optic nerve, and that passes through the optic disc. And you can't have photoreceptors there because you have to have a hole where the nerves can pass through, and that's what that is. So there's a hole in the back of the retina. That's the optic disc. The macula is this region here. This is where we start to have the highest visual acuity. So this is going to be the center of our field of view. Okay, and then we have the fovea, which is the small region at the back of the retina. And if you look at the back of the retina, you can actually see the fovea. It is the center of the macula, and this is specialized for very high acuity. This has the highest density of photoreceptors across the entire retina, so it's not like we have a consistent density of photoreceptors. It gets higher and higher the closer and closer you get to the fovea, and it peaks at the fovea. It has a very high concentration of cones. It has a low concentration of rods. There's a large absence of intervening cells as well. So let's talk a little bit about what that looks like in that proportion. So imagine, let me back up here. Imagine if we were to take a line and go across the retina, going through the optic disc and through the fovea like this. So we were just to take a line and we were to measure the density of rods and cones as we go across. This is what this figure is, okay? Except reversed. So as I said, the line going that way, we're looking at going this way. So basically, on the outer edge of the retina, which would be here and here, we have cones. There are cones that are out there, but there's not that many of them. We see a lot of rods. And then as we get closer and closer to our field of view, the density of rods increases, as does the cones, but you can see there's a lot more rods than cones on the outer edge of the macula, okay? Once we get into the macula, which is going to be right here, we start to see a transition happening. Now we get fewer and fewer rods, and we get more and more cones, to the point where the cones become very, very dense. To the point where we finally get to the fovea, where there are basically no rods at all, and it's entirely cones. So in this figure here, what we're looking at are field of views for rods, which are shown in purple, or cones. And as I said, like the cones that are on the outer part of the retina, there is convergence, which is why they have a large field of view. But as we get closer and closer to the fovea, and that's where most of our cones are located anyway, they're located right near the fovea, the field of view there for each cone is really, really tiny because it's the size of a single cone. And if you're looking at a cone from the top, it's not much. That's a very, very small profile. If you were to take that cell, if we're looking at it like this and we turn it like this, it's like looking at the end of a pencil, basically, right? So you're looking at a tiny little part. That's the entire field of view because we're getting one cone to each one bipolar cell, which is going to connect to retinal ganglion cells. So that information is really, really, that's where we have our highest degree of visual resolution. And that's the center of our field of view. And as we get further away, then we start to have larger fields of view for the cones. We start to have information from the rods as well, and for the stuff that they do. And then as we get to the outer part of the retina, now it's mostly dominated by rods, and there are fewer. There's less information that comes from cones. Although there's some, of course, right? Because obviously in your peripheral view, you can see color, but you don't see it at very high resolution, which is illustrated in this slide. So this is kind of showing how our peripheral vision works versus our foveal vision. Okay, so the macular the macular field of view. So our entire field of view covers a little bit more than 180 degrees. So if you're just staring at a dot, and then if you can envision the light information that's coming from the outer edge, it's a little bit beyond 180 degrees. You can see just a little bit beyond the edge of like your eye. That's what's illustrated here, right? So you've got far peripheral view, mid peripheral view, and the near peripheral view. But then if you're staring at that dot, if you just are staring at it, you notice something. If you like don't move your eyes from that dot, but you're trying to pay attention to the information that's in the peripheral, you can see that it's fuzzy. It's not sharp. And so what's happening is that we are directing our attention by moving where our eyes are located, and we're focusing on particular places within the field of view, and we're gathering the most sharp resolution of that information as the eyes are darting around. And we don't need that sharpness in the peripheral. We just need the sharpness in the center of our eyes. So there's no reason to have really high degree of a lot of photoreceptors across the entire retina, because it's good enough to have just a portion where we have sharpness. And that means the outer parts can do different kinds of calculations, as we're going to see. Okay. And so basically like this, if you were to stare at the eye here in the center, you can notice that like, okay, you can kind of, if you're paying attention to the center, you can see the sharpness of that A in the center. You might not necessarily see how sharp things are out here or out here as you stare at the A in the center. And this is because the visual system has to have attention. There's attention that is paid to it. Some of that is conscious and some of that is just unconscious. Our eyes are constantly flitting around, as we're going to talk about next time, that we make these saccades and they're constantly moving, and that the saccades are gathering information to create a mental image of what the room is filled with, right? I can only see at any one instant with a large degree of sharpness around 2 degrees of the entire field of view, so with really great sharpness. But as my eyes move around, they gather that information to create a total pattern. And we'll talk more about that next time. Okay. Crap. Running late. I might have to cover some of this in lab today, which is good. So we have lab. So, okay. So that laminar organization we've been talking about, we've got the ganglion cells on the outside, we've got the photoreceptor cell on the inside, and then we've got these bipolar cells and horizontal cells in the middle. Now, one thing about the way ganglion cells, the way ganglion cells work is that they fall into many different categories. There's lots and lots of different ganglion cells. We're just going to talk about 2 of them, and these are 2 that are opposites that fall into the same kind of category. So ganglion cells, there's a lot of processing of information that happens within the retina before it even gets to the rest of the central nervous system. That processing of information, some of that can be, do we have light or dark opposing to each other? Do we have light in the middle versus dark? And this is referring to the center and surround. So the center may be a cone or a rod, but typically we're going to be talking about cones. And then the surround is also going to be a bunch of cones. And if we're referring to one type of ganglion cell, we're going to start with this. This is referred to as an off ganglion cell. Okay, so these ganglion cells, right, these are located here. They are true neurons because they actually have action potentials. If we're measuring what an off ganglion cell is, the way we get an off ganglion cell to fire is that if we decrease the intensity of the light in the center of its field of view, okay, of its receptive field. So it may have a receptive field for which if we change the intensity of the light, we can cause it to do stuff, okay? And this sort of makes sense, right? I told you that that as we that if light hyperpolarizes a photoreceptor, that if we turn down the light, that means it's depolarized, and you can envision that it's going to release more glutamate, and it does, right? I said that. So if we're decreasing the darkness here, so if we decrease the amount of light, we increase darkness. What happens to an off ganglion cell? It increases its firing rate. So its firing rate is telling us that the center of this field of view is darker than the outside. So there's darkness here, and then it's lighter out here. And the reason why it's an off center is because if we increase that dark patch, so if we decrease the amount of light past the center, then we actually get a decrease in its membrane, in its firing rate. So if it's really, really bright in the center, its firing rate is really quite low, if we're talking about an off-center ganglion cell. If we turn off the light in the center, it increases its firing rate. And then if we increase that dark patch that we're shining to that center of its receptive field, it actually increases. This is why it's referred to as an off-center, because in the center of its receptive field, if it is off, if the light is off, that's going to be the thing that it's going to elicit the most action potentials, the highest rate of action potentials. So an off-center ganglion cell means the off is turned, so the center is turned off, and that's going to make that ganglion cell fire. Okay, and the way this works is that, so if you can envision that this is the kind of light of the receptive field for this particular cone. So we've got this cone here, and if it is attached to an off-center bipolar cell, so some of what determines whether we're talking about an off-center ganglion cell is whether it's attached to an off-center bipolar cell, which has its own unique physiology in that it has AMPA receptors. And remember, we know AMPA receptors, they're sensitive to glutamate. And if glutamate is being released by this photoreceptor, what happens is that, basically, if we have light turned off, imagine if we're turning off the light here at time 0, so T0 is here. If we turn on the light in the center of an off-center ganglion cell, so we turn on the light, we are going to, let's say we're measuring the membrane potential of the cone. The way you can track this is that the cone is green, we're looking at green here, this is sort of bluish purple, this is here, and then now for measuring the activity of the off-center ganglion cell, we're looking at it here, okay? So an off-center ganglion cell, if we turn on the light in the center, we hyperpolarize the cone, as we've talked about, right? Then it means that there's going to be less glutamate released at this synapse, which means that this cell is also decreasing its membrane potential, right? So if there's less glutamate hitting here, this AMPA receptor, is going to get less glutamate, so therefore its membrane potential decreases. So as soon as we turn on that light in the center, it decreases, which means that this cell is no longer releasing glutamate as much, and therefore the off-center ganglion cells has less glutamate at its synapse, and therefore it decreases its firing rate. And it can even turn off if you have, especially if you have the light on in the center and dark on the outside. And then as soon as you turn off that light, then we get action potentials returning, because it's an off-center ganglion cell. It likes it when the light is turned off. So if we turn on the light, you decrease the firing rate of an off-center ganglion cell, and this is the series of things that has to happen in order for this to occur. So if we have a dark spot, so let's say we've got a field of view, but then we decrease the amount of light that's hitting the center of the field of view, what we see is an increase in the membrane potential, right? Because these photoreceptors they're graded in their response, right? So if we decrease the amount of photons that's hitting this cone, then it's going to depolarize. It's going to be releasing more glutamate at its synapse, right? Because it's done in a graded way, which means it acts on these AMPA receptors, causing greater depolarization in the off-center bipolar cell. That off-center bipolar cell increases its membrane potential, which means during this phase between T1 and T2, it is now releasing more glutamate at this synapse, which increases the firing rate of the off-center ganglion cell. So as we turn the light off here, we get firing happening. As we turn up the light, we can decrease the firing. So you can see that this is graded, but what's really changing is the number of action potentials. So we're changing the firing rate. And this is an off-center ganglion cell.
Since we're talking about an off-center ganglion cell, obviously that must mean that there's an on-center ganglion cell. So let's talk about that. So in this case, what we're looking at is an example of a recording from a ganglion cell that's an on-center ganglion cell. What it means is that if we turn on the light in the center of its receptive field, that will end up increasing the action potentials coming from that on-center ganglion cell. So let's walk through the different steps here. So if we have a relatively dark illumination provided to the entire receptive field of this particular cell, we're going to have a certain sort of spontaneous firing rate. As we increase the brightness in the center of its receptive field, this will result in an increase in the firing rate of that on-center ganglion cell. It likes it when light is turned on, therefore it's an on-center ganglion cell. But the other interesting thing is that as we provide more and more light to the surround, this results in a reduction of action potentials relative to the peak. Now, in this case, it still might be relatively high compared to when there's no light on at all. But there's definitely a reduction relative to when we're just illuminating the center. That's because the surround provides inhibition, as we will get to in a moment. But first, let's just talk about how the center works. So in a non-center ganglion cell, we have a cone here located within the center of this receptive field. You can just imagine this is a conceptual receptive field. We've got the surround, we've got the center here. And what we can see is that this on-center ganglion cell is connected to a different kind of on-polar bipolar cell. This on-center bipolar cell expresses not AMPA receptors, but a metabotropic glutamate receptor. That when glutamate binds to it, it actually ends up hyperpolarizing the cell. Now, typically, we think of glutamate as being an excitatory neurotransmitter. And indeed, it is with off-center bipolar cells, like we were just looking at before. But in this case, glutamate, as it binds to the metabotropic glutamate receptors on the postsynaptic side, it actually ends up with a decrease in the overall membrane potential when it binds. When glutamate binds, it induces a hyperpolarization event. And that means that, of course, this on-polar bipolar cell, if it becomes hyperpolarized, it results in a reduction in the amount of glutamate that's released onto the on-center ganglion cell. So let's talk about that. So we'll start over here with the dark spot in the middle, right? So we've got T0 here, and then we have T1 when we decrease the intensity. So we have a relatively dim illumination applied to the entire receptive field, but we can reduce the luminescence even further in the center. So when we reduce that luminescence even further, what that does is that it depolarizes the cone, right? Because light hyperpolarizes and darkness depolarizes it. So that means it releases more and more glutamate onto its on-center bipolar cell. But there's an interesting thing that happens in the postsynaptic on-center bipolar cell. Because glutamate acts as an inhibitory neurotransmitter at this particular synapse, it then results in a hyperpolarization of that on-polar bipolar cell. It becomes, less polarized, and therefore it releases less glutamate at its synapse, which means that it reduces the synaptic drive onto the on-center ganglion cell, and therefore the on-center ganglion cell reduces its firing rate. And if it's low enough, it will result in no firing whatsoever. If we turn on the light in an on-center ganglion cell, what we see, of course, light hyperpolarizes the cone, and therefore it releases less glutamate at the synapse, and that means that there is less inhibition being placed onto this on-center bipolar cell. And so therefore, there's lower inhibition. We've removed the inhibition onto the cell, so it's become disinhibited, and therefore it becomes depolarized. And since it is depolarized, it releases more glutamate at the synapse, which means it increases its firing rate. The on-center ganglion cell increases its firing rate as shown here. So this means that a hyperpolarization of the cone gets flipped because of this inhibitory synapse. And that's the interesting thing about inhibitory synapses, is that they can result in an inverse of the kind of activity that's flowing through the network. So let's compare these two pathways, looking at them together. So we've got one cone connected to two different bipolar cells. One is an on-center bipolar cell, one is an off-center bipolar cell. The on-center bipolar cell is inhibited by glutamate because it is expressing a metabotropic glutamate receptor. The off-center bipolar cell is excited by glutamate because it has AMPA and kainate receptors as well. And this means that because we have these two different synapses that get affected in opposite ways, it means that we can get the inverse happening with the same kind of activity happening within the cone. So when light is turned on in the cone, we get hyperpolarization of the cone. There's less glutamate released here. This cell becomes disinhibited, so it becomes depolarized and increases the firing rate of its postsynaptic ganglion cell. And therefore, when light is on, it is firing, and it is an on-center ganglion cell. In this case, we have less glutamate here, which means that this cell becomes hyperpolarized, which means it releases less glutamate here. And the off-center ganglion cell turns off when the light is on. And then, of course, we get the inverse when the dark turns on. Now, the way surround works is also through cones. What you have to imagine is that a cone just isn't sitting in isolation, right? A cone is going to be in the retina. It's going to be loaded with next to a bunch of other cones. And so there's a bunch of cones around here. So not even just two. You can imagine that there's probably like 10, 15, 20, a bunch of cones that are within the surround. And they all have their own receptive fields. But for this single cone that we're focused on in the center here, this cone has its, there's an on-center bipolar cell and an on-center ganglion cell attached to it, which means that if there's light that's being presented to the center of its receptive field, it increases its firing rate. As we're seeing here, this is the number of action potentials happening per second. The spontaneous level of activity is shown here. So this neuron, this on-center ganglion cell at the basic level of illumination apparently prefers to fire at around 55 55 impulses per second. But if we shine a little bit of light, even just a little bit of light in the center, we can increase its firing rate substantially to something like, 88 action potentials per second. So that's what this is doing here. And then as we move the light away from the center of the receptive field, so we get further and further away. So right here, we're nearly at the edge of the light. That's what this is indicating, right? At the edge of the center of the receptive field. That's when we start to see it dip a little bit. And so now here we're looking at light that's being presented to a little bit of the center and a little bit of the surround. And we can see we're getting really, really close to the spontaneous level of activity. But as we move the light all the way into the surround and none of the light is in the center, we can actually see that the spontaneous firing rate gets below the we can see that the firing rate that we're measuring is below the spontaneous level of activity seen in this neuron at this point. This is also true here because the illumination is being provided to the surround and none to the center. And then eventually, as soon as we're out of the receptive field for this entire complex, well, what we see is that the firing rate then returns to its spontaneous firing rate. This is because nearby cones provide lateral inhibition to whatever cone is in the center of the receptive field that we're recording from. So this cone is providing inhibition to the cone in the center, and this cone is providing inhibition to the one that we're recording to in the center. So if there's illumination being provided to the surround, well, it turns out that the horizontal cells that are adjacent to them, these horizontal cells, that's the type of cell that's providing horizontal information across the lamina. It is inhibiting this synapse, and it's resulting in a decrease in the activity of the on-center ganglion cell. Another way to illustrate that is shown here. So if we provide just a little bit of light to the center of the receptive field of this neuron, of this on-center ganglion cell, we'll see that it has a firing rate that's relatively high. As we increase the size of that light that's being illuminated to the center, we can see that it's just going to get brighter and brighter, and that's going to result in some kind of maximum rate of firing rate within the on-center ganglion cell. So it's going to reach peak at some point. But then as we increase the size of the spot, the light spot that we're providing to the center. And as it starts to take on some of the surround, we see something interesting happening. We see that the firing rate actually decreases. So even though we're providing more and more light, the firing rate actually goes down because now we're starting to include some of the surround. So we're starting to illuminate some of the cones that are in here, and now they're starting to inhibit the center cone. So the center cone is at its maximum, right? There's as much glutamate that could possibly be released here, but some of it is being inhibited because it's not able to release as much glutamate as it normally would because of the inhibition being provided by the horizontal cells, which means that there's less of there's less disinhibition of the on-center bipolar cell, which means that there's less glutamate at the synapse when we start to include more and more of the surround. And then as we illuminate even more of the surround, now there's even less glutamate at the synapse because of the inhibitory effect that's being placed upon uh, the the junction between the center cone and the on-center bipolar cell. So, okay, hopefully that clarifies aspects of how lateral inhibition works to the center and the way horizontals actually provide that lateral inhibition, and also that you have a greater appreciation for how one inhibitory synapse can actually lead to the inverse of a signal, where we can then have downstream neurons firing opposite to if we didn't have that inhibition. So if you like this kind of content, please like and subscribe, and I will talk to you later. Thank you.